Bacteria must capture energy from food sources in order to grow and reproduce, or, in other words, to stay alive. When we discuss bacteria capturing energy, what we are really talking about is the ability of bacterial cells to use a chemical substrate, a food, to generate ATP. ATP is the high-energy molecule used for storing energy and building all the necessary cellular components. In anaerobic environments, bacteria can capture energy and store it in ATP using either anaerobic respiration or fermentation. This lesson will introduce fermentation. Fermentation happens in a wide range of environments, but in case you think you personally don't care about fermentation, just think of the last time you had a refreshing beer, a creamy yogurt, a fragrant cheese, or a warm slice of bread. The process and products of fermentation by bacteria and yeast are responsible for the rich flavors and textures encountered in these foods. Fermentation is the anaerobic catabolism of a single chemical compound using a series of redox transformations with the goal of generating ATP by substrate-level phosphorylation. That definition sounds complicated, so let's break it down a little bit. Catabolism just refers to the breakdown of a chemical compound. During fermentation, the breakdown occurs through a series of redox transformations, which is just a fancy term for electron-donating and electron-accepting reactions. All of the redox transformations that take place act on atoms found in the original substrate molecule. Some atoms will act as electron donors and some as electron acceptors. The electron shuffling requires a series of enzymes that accelerate the reaction and catalyze the production of various intermediate forms of the initial substrate. Through these fermentation reactions, the energy present in the chemical bonds of the substrate can be captured to produce ATP by substrate-level phosphorylation. Substrate-level phosphorylation is the production of ATP by transferring high-energy phosphate groups from an organic molecule to ADP. Let's look at the classic example of fermentation, glycolysis. Glyco means sugar and refers to glucose, and lysis means to split. So glycolysis is the splitting of glucose. We will call the first stage of glycolysis the prep stage. In this stage, 4 enzymes are used to prepare glucose for the rest of the pathway. Glucose is split into 2 molecules of glyceraldehyde 3-phosphate. In the prep stage, there are no redox transformations yet, and no energy has been released or captured. In fact, energy is put in at this stage to transform the glucose. In the second stage, this is where the important action happens. This is where the redox transformations take place that lead to generation of ATP. First, an enzyme adds a phosphate group to those 2 molecules of glyceraldehyde 3-phosphate from the prep stage. These phosphate groups are high